Hi, this is our DNA Blast Lab.
So for introduction, we had the key vocabulary. The key words were human genome project, the genome, the bioinformatics, the blast, the cladogram, the common ancestor, the derived characteristics, and the cladistics. For the questions, the first question was how is information from the Human Genome Project used in bioinformatics? And we answered it that the information from the Human Genome Project provides various tools and programs which are used to search the genome of thousands of organisms, including the human genome. All the data collected is then used in bioinformatics to provide more knowledge and information to research already there. The second question was what kind of information is obtained from a blast search and how it can be used. And our answer was the information obtained from a blast search is that you find the similarities between biological sequences, and the information is used to compare certain gene sequences for any type of research being done. The last question was how are cladograms that are currently being created different from cladograms that were created in the 1970s? And we answered that the cladogram that are currently created are different from the cladograms that were created in the 1970s because it wasn't until the 1970s that cladograms were called cladograms. They had a different name, which was phylogenetic systematics, and then Henning intended his method to construct phylogenies, but in 1965, Ernest Mayr started calling them cladograms. Oh, cladistics, I mean.
Okay, so these are our cladograms. This is our hypothesis. We decided to place the fossil right here, like under crocodilians. The fossil shows evidence that the organism had characteristics of both crocodilians and birds. This is cladogram number 2. We placed our gene number 1 was right here under birds. Gene number 2 was above vertebrae. Gene number 3 was below rodents, and gene number 4 was below crocodilians. And then cladogram number 3. Oh, well, this is just it. In conclusion, our hypothesis was supported by our third cladogram. After blasting the genes, it showed that we placed the unknown organism correctly. The data showed the fossil is very closely related to birds and also a common ancestor with crocodilians.
And this is our fourth cladogram. The gene we decided to blast was keratin, and we found that the closest species to the human keratin were the gorillas, orangutans, and chimpanzees. And this is our gene rank. For the first one was collagen, and we ranked the Shaker gene 2, the ubipintin conjugating enzyme 3, and the sinuses mitochondrion 4th. And this is our bibliography. Thank you.